So I will begin. So first, hello everyone, I'm Yiran, I'm the technical support manager from Yiran. So firstly, I want to use thanks for inviting me to this webinar, and it is good to meet you all with this webinar. And today I will try to give a clear and brief introduction to NGS and library preparation. So before I begin my presentation, please allow me to give a brief introduction of our company, Yiran. So Yiran is a compound of two words, innovation and enzyme, which means innovation in enzyme technology. We are a supplier of life science, R&D, and biomedicine with independent R&D labs, production lines, and the network of global distribution. We have always been providing support to the scientific research. Our products are widely used by some universities and institutes. We have been mentioned in CNS and sub-journals for over 180 times and more than 500 times in various other journals. So far, the total citation of papers has reached to 10,000. And I will talk about my presentation in five aspects. First, I will give an introduction of the library preparation, and next I will move to DNA library preparation, and then RNA library preparation, and the next is epigenetic library preparation. And finally, I will talk about the library quality control and the cleanup. And first, let's begin with the introduction of library preparation. The total NGS pipeline includes the library preparation, sequencing, and data analysis. And today, I will focus on the first step that's called library preparation steps. And there are several library preparation strategies. There are DNA sequencing, RNA sequencing, single-cell sequencing, and epigenetic sequencing, and the quality control strategy for the library preparation. Here is the total library preparation pipeline. Firstly, we will begin with the DNA or RNA sample, and then the intact DNA or RNA will be break into the accessible length for the sequencer. And then the adapters is adding to both ends of the fragments. The adapters contain the oligos, which is complementary to the sequence on the sequencing chip, which also called flow cell, and also contains the common primer pairs for the DNA synthesis. Synthesis. And the steps aimed for the massively parallel sequencing. And the next step is the size selection. These steps use the base to select the appropriate size range of the fragments, because there are some unwanted fragments, such as primodamas, exist in the sample, so which could cause bad sequencing quality, and hence it is need to be removed from the sample. And then we will do the PCR to amplify the library to increase the library yield. And finally, the cleanup will be done to purify the library. After that, the library is ready for sequencing. And okay, let's move to the DNA library preparation part. There are two different fragmentation methods for DNA library preparation. They are physical fragmentation, enzymic fragmentation, and endemic fragmentation. So physical fragmentation is an additional physical sharing machine, which is very expensive, and some labs may not equip that. Compared to this method, the enzymic fragmentation method is much cheaper and do not need additional equipment. There are two main methods for enzymic fragmentations, and that are the first method is based on the endonucleotide method, and the second method is based on the transposes. And there is a trade-off between the randomness and the wastage of samples. So when your sample is very pressure, you had better to choose the endemic fragmentations for its low wastage. And here is the steps for the physical fragmentations based on the DNA library preparation. The physical sharing machine is used to break the intact DNA into small pieces. And then the unrepaired DNA tolling adapter ligations are done to add the adapters to both of the fragments. This method can give less bad libraries because it's based on the physical fragmentations, but it needs additional equipment and need to expensive consumable. And here are the steps for the endonucleotide fragmentation-based DNA library preparation. The endonucleotide is used to break the intact DNA into small pieces, and the fragmentation and the repair detailing can be combined into one step. And hence, this method is very time-saving and really shortens the hand-on time, as it is used in enzyme to do the fragmentations. This method can be done with the standard lab equipment, no need for additional physical showing machine, and the enzymic fragmentations can give lower sample loads, and this method is appropriate for the pressure sample. And here is the DNA library preparation pipeline based on the Transposes method. This is the new method which do the DNA library preparation. The picture on the upper right is the structure of the Transposes, which also called TN5, and this enzyme is preloaded with adapters. During the fragmentation step, the TN5 can ligate the adapters while fragments the DNA at the same time. Since the TN5 combines so many steps into one step, this method is easiest way to do the library preparation. And normally the whole process can be done within 80 minutes. And this is really time saving. And this method can also be done with the standard lab equipment. Compared to the endonucleotide method, the Transposes method is less sequence specific, hence can generate less biased library. And the method above our aims for, and the method above generates the libraries contains all the genome DNA, and sometimes we only need to sequence on the specific region, hence we need to do the target sequencing. And compared to the whole genome sequencing, the target sequencing is rapid and cost-effective way to detect the known and novel variants in the selected set of genes or genomic region. There are two main methods for the target sequencing. For the sequencing of the larger gene content, for example, the whole exome sequencing, we can use a probe to capture the target sequence we want after the library preparation steps for the whole genome. And for the smaller gene content, for example, such as the cancer-related genes, we can use the multiple PCR to amplify the target first, and then use the library preparation kit to finish the library preparation. And here's the pipeline for the applicant sequencing. It is begin with the total DNA, and then we use multiplex PCR to amplify the region we want, and then we digest the primers, and then lab steps are the same as the library preparation steps for the whole genome. And based on the different fragmentation method, Wisdom offers different DNA library preparation kits to meet the different requirements. We have kits compatible with physical showing machine, without physical showing machine, and also have kits for the target sequencing. So here are the kits based on the physical showing machine, physical fragmentation method. This case is used after the genomic DNA fragmented by the physical showing machine. And this case includes end preparation, adapter ligation, and library amplification modules. From the product name, we can see the biggest trait for these two cases is universal. It is applicable for the broad range of input DNA and extensive species. Both kits can give high quality and high yield sequencing results. And the difference between these two kits is that the later one comes additional DNA damage repair module, which is specially designed for FFPE sample, which is commonly used in hospitals to preserve the tumor sample. However, during the progress of making FFPE sample, it will cause damage to DNA, which could lead to the false positives and in the downstream analysis. Hence, it is necessary to do the extra DNA damage repair steps for the FFPE sample. And here's the test result of our case compared to the competitors. It shows that our case have good performance. And next, I will introduce the case based on the endemic fragmentation method. And this case contains the additional endonucleotide module that can break the intact DNA into small pieces. And it combines the fragmentation, and repair, and detailing into 1 steps. So it is easy to use and very time saving. And Wisdom also offers library preparation kits based on the Transposes method. And this kit come prepared library within 90 minutes, and it is very time saving, and it is universal and applicable for the variant amount of input DNA from 1 nanogram to 15 nanogram of input DNA. And from the test result, we can see this kit has good performance compared to the same product from Illumina. And this kit has been widely used by the scientific research. You can see here are listed selected citations in recent years, and they are from the top journals such as Cell, Nature, and Nature Medicine. Also, this kit can also be used in ATAC sequencing. So ATAC sequencing is the abbreviation of assays of transposes accessible chromatin within hash output sequencing. The chromatin compactness has been considered a major determination of gene activity. It is divided into 2 states, the closed chromatin and the open chromatin. Tightly packed closed chromatin is considered as a hallmark of gene sequencing. The open chromatin is considered as an excellent indicator of the gene expression. The transposes TN5 is used to segment the open chromatin region and then capture the DNA, and then the captured DNA is then sent for sequencing. And here's a kit for amplicon sequencing. It is ideal for the mutation detection and target on the target region. And the results shows that our kit has good performance as good as the sample feature, and this kit is capable for the Illumina and Torrent capture panels and the sequencing platforms. And here next we will move to the RNA sequencing library preparation. The RNA sequencing is usually done to look at the gene expression to identify which gene is upregulated or downregulated in a given sample. Compared to the DNA sequencing, the RNA sequencing is a little bit, the library preparation steps for RNA sequencing is a little bit different. After RNA extraction, we will do Rna enrichment to remove the undesired type of RNA, and then we will use high temperature and high salinity to break the intact RNA into small pieces. And then the next step is to convert RNA to DNA. That step is called cDNA synthesis because the sequencers only works for the DNA, so we cannot sequence RNA directly. And the other steps are the similar as the DNA library preparations. So why we need to do the RNA enrichment first? It is because most of the time we are interested in the messenger RNA. And but after RNA extractions, most of the RNA are the ribosome RNA, so they are not wanted from the downstream analysis. So we need to do the enrichment first, and there are 2 ways for the RNA enrichment. First is the mRNA capture strategy. That method uses the advantage of the fact that messenger RNA has poly-A tail on the 3' end. We can use poly-A to capture poly-A capture techniques. And that is a base which has oligo DT on the surface. Those bases are all able to capture the messenger RNA through the interactions with poly-A tails, and then we can elude those messenger RNA from the base for downstream steps. Instead of capture messenger RNA, we could delete the ribosome RNA, and that strategy is called the RNA depletion strategy. And there are several ways to do this, but the most common way is to use a panel of probes that will bind to the ribosome RNA, and then we use enzyme to digest the probes and the ribosome RNA. And after that, the left RNA can be used to go through the library preparation steps. And for the cDNA synthesis, there are 2 ways to do that. After RNA are fragmented and the enrichment, and the synthesis of the first strand cDNA, we can choose whether we want to build the stranded RNA sequencing library or non-stranded RNA sequencing library. For the stranded RNA sampling library, we only want to keep one strand of the cDNA, and that need us to add DUTP instead to add DUTP when we do the second strand synthesis. And then we need to remove U after the adapter ligations and set selections. After that, we only left the strand which comes from one strand, and then it can be sent for sequencing. And WISAMS offers the mRNA enrichment solutions and also the library preparation kits for the non-stranded transcript library and the stranded transcriptome library. And now I will move to the epigenetic library preparation. For the epigenetic, we focus on the binding focus on finding the interactions between the protein and the DNA. And there may be some transcription factors, histones that bind to DNA could influence the gene expression. And the classic sequencing way for finding the protein-DNA interaction is called CHIP, and it begins when the entire cell and then goes through the cross-linked cell lysis, antibody binding, immune approbation, cross-link reversal, and DNA cleanup, and then finally we do the cleanup, and then the samples are ready for sequencing. And this method required high initial cells, so it is not compatible for the pressure samples, and it is based on the physical sharing machine, and the experiment period is very long, so it is complicated. So the whole total experiment process is complicated, and the repeatability for this method is also very poor, which means the result is not consistent from each trio. And the sequencing result for this method has high noise, which will cause problems to the downstream analysis. And in recent years, there is a new method to do the protein-DNA interaction analysis that is called cortex sequencing. And it is specific technology that designed for the protein-genome interactions that can seamlessly replace the traditional chip sequencing. And this method is based on the transposes, and it begins with a small amount of cells. So it do not need a huge amount of samples. And then it uses the primary antibody to target the protein we want, and then use the secondary antibody to enlarge the signal. And then the transposes are recruited to cut the segments near the target protein and add adapters to the both and all fragments. And then the DNA can be extracted, and after that, the PCR were used to isolate the sequence we want. And the whole process can be done within 1 day, and do not need additional physical sharing machine. Hence, it is easy for us to do this whole experiment. And this method can give lower noise compared to the chip sequencing, and it has high repeatability, which makes the downstream analysis much easier. And WISAM offers the case for the cortex sequencing, which makes the sequencing of protein DNA interaction much easier. And we also offer the technical support during the whole process. You do the cortex experiment. And here I list one select paper that uses our kit for target sequencing, and it's also from the top journal. So it proves that our kit has good performance. And now I will move to the additional materials that also required in the library preparation, that include the DNA or RNA quantification, quality control, and clean beads. So there are three main methods for the library quantification and quality control. And the first one is based on the qubit, that is for the total nucleic acid quantifications. And the second method is based on the bioanalyzer, and it is aims for the library set distributions. And the final and the third method is based on the qPCR, and it aims to quantify for the valid library. And we have solutions support both qubit and qPCR method during the library quantification and quality control. And for qubit solutions, we have a qubit product to meet different qubit product series to meet the requirement for different qPCR equipment. And we also offered, and the CleanBase is also used for the library cleanup and set selections. During the whole process of the DNA or RNA library preparation, there may be some contaminations contents in the sample. And there are also maybe some fragments we do not want, such as the prima that is small fragments we don't want. So we need to remove it by using the CleanBase. And here is the structure of the CleanBase. And we can choose which side we want by change the ratio of the base. And we also have, and this base is applicable for the DNA cleanup and set selection in NGS library preparation. And our product with them DNA CleanBase is perfect alternative for the Beckman's Ampere XB base. And the usage for this base is the same as Ampere base. So you can directly change the Ampere base into our base without changing any protocol. And our base is much affordable. And yeah. Okay, that's all. Thanks for your attention.